and method of discovering drugs. One of the important ways is research. Research is inquisitiveness. Try to know what such a particular thing does. But one thing must be acknowledged. For one to be able to discover a drug for a particular illness, you must know the pathological sequence of the type of ailment you are developing drug for. Yes, the clinical cause and variation, developmental stages of such disease, the causative factor, and many more. Having such knowledge, you can now be ready to do a lot of research on various types of materials. Yes. You see, the synergism of drugs, what I mean synergism, combination of many drugs to yield a particular goal matters a lot. Ability to do that matters a lot. But for you to have that ability, you must have in-depth knowledge of disease formation, any clinical cause, the pathology of diseases, the developmental stages, ways in which such disease spread in the body. If you don't have such in-depth knowledge, the potential drug such individual is developing may have a command. And as such, you will end up not developing a proper drugs. Yes, for instance, you're trying to develop a drugs, maybe for viral-based diseases. The materials that will come to mind to be used for the development must be material that will not injure the cell lines. Apart from knowing the pathological indices of the body, the physiological well-being of the body must also be acknowledged. You must have that basic knowledge. Because after development of the drugs, you must do a cytological, histological assay to check if there are cellular changes. A drug could be effective, but if there are cellular changes, maybe some cells are damaged in the body, such drug could harm any person that one may administer such drug for. Even the biochemical parameters must be in place. Yes. There are some of these inflammatory diseases, especially some few bacterial infections and many viruses. You see, viruses are difficult to treat. There are some materials found or some elements found in some foods that when isolated will have effect on such viruses. Yes. You see, there are some materials, for instance, let me just give an example. Food that we eat, there are some food that are acidic in nature. Yes, that we eat, but the body tolerates such acids. And there are other materials that one could capitalize on, synthesize it, and modulate it maybe with other material from other food stocks, herbs, or it could even be minerals. Then administer it to such individuals that have such problem. Normally, most times, animal trial, we do animal trials. But in a situation whereby disease is only limited to humans, there are always volunteers. First, one have to, it cannot develop what I would call exogenous trials, not endogenous trials. Why I call it exogenous trials? One could exfoliate blood from individual and do experimental trials on those blood. First, before endogenous, what I mean, not removing blood, trying the drug directly with human being system, volunteers. Yes. For instance, the issue of HIV is only related to human being. And so you can only do HIV trials then. You are trying it on apes or animal. No. Because most of these animals don't have such a problem. Some of them would develop with their immunological system combat such disease. I have done a lot of trials on that. So you remove blood and try to treat the blood, infected blood. In the course of the treatment, you do a kind of biochemical assay and histological and cytological assay. What I mean cytological, I'm talking about trying to test the cell, whether they are degenerated, whether there is structural abnormalities, and all that. It is very simple, but it has to do with high-level reasoning. High-level reasoning. Having background knowledge of pathology, the indices of pathology, clinical cause of a disease, pathological consequences, and all that. So you'll be able, first of all, in drug development, you assume certain materials to be effective on the disease you are working on. First of all, it's based on assumptions, yes. As you know, viruses are intracellular. And so if you use certain an agent that could maybe have effects when you are tackling virus, you use indirect to tackle. You don't just go directly, otherwise you may affect the cell lines of the body. And when the cell lines of the body is affected, even though the virus is dead, but the cell line is also dead, the individual dies. Nothing is achieved. There are ways you could stimulate immune responses, stimulate immunity, so that certain immune cells will be strengthened to combat such viral infections. Yes, and there are certain basic foods, some of them that contain acids. The food we eat, there are some that contain acids. But I don't want to mention them now, right now. And so if you are able to, to synthesize those things, add other nutrients, because drug development is merely synergism. Synergism means you combine this one, bring this one to see how it will be powerful against the disease you are trying to treat. Because it's just experimental trials. But if you did not even try, there will be success. When we say research, it's not just talking about, at school we say research methodology. Who that designed such a program or course. It's not just title of you. If you ever say, the title of your subject matter, and then abstract, review, literature review, discussion. No, that is not research. When we say research, research is reasoning, agitation within the mind and the brain. Try to break through into the unknown, into impossibility. You are taking step out of ordinary. People with high reasoning, reason ahead of time. Now, it could be maybe a common thing that we are using, we use daily, maybe for dietary purposes. You know what that stuff is used for. You saw another thing. The mind will now be agitating. Should I do this? If I combine this, will it work? Yes, it may work, because this is how this disease is being developed in the body system. You will just be talking to yourself, then start bringing all those things together. And that is the same sequence engineering program, engineering technology is developed. Once you always learn to take a step out of ordinary, everything is based on reasoning, research, and experimentations. If you do not experiment, you will get results. Yes. And if you drugs are be able to develop, it was how the outcome came to pass. Yes. First of all, knowing the pathology of a disease matters a lot. For instance, issue of asthma. I already know the pathological sequelae that is a respiratory airways diseases. How certain mucus can just on its own be secreting and blocking the respiratory tree. And how it could, on the long term, affect the alveoli, which are the cellular unit, the parenchyma of the lungs. And how certain atopies, Mansell histamine, are involved in such formations and all that. Then, an individual, a researcher will look inward. Is there anything unknown that will not revealed by textbook? Because scientific laws or medical laws were discovered by humans, and humans, there's always a micro error. Yes. When you start thinking this way, then what are the things one will do? What are the herbs? Because all drugs are made from herbs. It's either you sit inside the active ingredient, or you produce it as herb. So, having known that, and having wide knowledge in foods, in therapeutic nature of foods, herbs, minerals. When I say minerals, it could be soil, it could be anything from the earth. Some of them have therapeutic purposes. This is the method in which drugs develop. And what are the likely side effects? For instance, individual is having severe headache, and you wouldn't know whether that severe headache is due to hypotension, low blood pressure. Low blood pressure is different from hypertension. Somebody that is clinically inclined, that has basic knowledge in medical science, and that have experience, should be aware that somebody with low blood pressure, when you give an analgesic, common paracetamol or any other pain-relieving drug, such problem get worsened. Because analgesic in itself has a mild effect on BP. It's not a BP drugs. It's a pain-releasing drug. So all analgesic, when I mean analgesic, all pain-relieving drugs are mostly administered to people with normal BP and high BP, not people with low BP. That is why you see in most hospitals, physicians, doctors that are not experienced, they give ordinary paracetamol a normal dose and the person collapses. And they claim it to be idiosyncrasy. It may not be idiosyncrasy because such doctor doesn't have in-depth knowledge of pathology or experience. Pathology is is a medical course. The fact that you study it does not mean that it will just be so grounded. It will become grounded through practices, through practice, Yes, that is how it is. And a lot of experimental trials that have taken place, that so common drug was given to so person, and the person collapsed, either fainted or died, especially paracetamol. So these are the cardinal area that one should look into. So what I'm trying to tell you is that in drug development, so many things must be registered in mind in the course of development of drugs. Because drugs are not food. And you must be able to know the adverse effect of that drugs. All drugs has adverse effect. That is why some drugs go through experimental trials. You go through biochemical trials, go through competent trial, whether what you claim the drug treat actually works, and the drug should be free from microorganisms so that when you administer the drug, the individual will not be infected. And also go through histological trials to check whether in the course of you developing that drug or when you administer that drug, whether that drug injure the tissue or cell lines of the body. It's only when such drug go through all the trials successfully that such drug will be regarded as a pharmaceutical product. It's simple. It's just for us to make up mind for development, to help humanity. Thank you very much for listening.